Here you go.
Welcome everyone to Stability Hub's webinar, ICHQ1 Guideline Revision, Strategic Implications for Stability. Our sponsor today is Q1 Scientific, a Cambrex company. Our presenters today are Aideen Glynn, Quality Manager at Q1 Scientific, a Cambrex company, Daniel Kirschner, Executive Director, Analytical and Biopharmaceutical Service at Cambrex. Our panelists include Sherilyn Campy, QC Head at Accord Healthcare, Walter Routh, founder of Sagana Consulting and associate editor at StabilityHub, and yours truly, John O'Neill, founder of the Pharmaceutical Stability Discussion Group and editor at StabilityHub. A few housekeeping notes. It adds a lot to the event if you can turn on your camera. We can deal with passing children and cats, and it's fine to be having your lunch while muted. Participants should stay muted unless invited by the moderator to unmute. Questions and comments can be submitted via the chat feature. We also conduct polls through the Zoom raised hands feature shown on the attendance roster. Just be ready to quickly remove them as soon as the count is taken so they don't throw off a later poll count. This meeting is being recorded and will be posted in the articles and videos section at stabilityhub.com. The monthly newsletter article of the same title is available in the site blog section if you care to do more detailed reading on this topic. With that, we'll turn the program over to Aideen and Daniel. I encourage you to submit your questions via chat at any point in the presentation as they pertain to the slide or topic being covered. Aideen and Daniel, presentation is yours.
Hello, everybody, and welcome to today's webinar on the ICHQ1 guideline revision and strategic implications for your stability programs. So what can you expect today out of us? Well, we're going to have a 40-minute presentation looking at the background to ICHQ1, key insights, what are the updates, and what is the industry feedback. We'll present some practical takeaways, what you need to do to prepare, and we'll have some questions that we will discuss out with the panel. As John said, ask your questions anytime, use the chat box throughout, and we'll address as many as possible as we can. Please engage with us, give us your feedback, tell us your thoughts. This is all about collaboration and like-minded individuals coming together. So just a bit about me. So I'm the quality manager here in Q1 Scientific in Waterford in Ireland. I lead QA and regulatory compliance across GMP and GDP stability storage sites. And we have sites in Ireland, in Belgium, and in Durham in the USA. Personally, I have over 18 years in the pharma industry, mainly involved in stability programs, audits, and regulatory strategies. I've held former senior roles in Airgen, Nipro, Newport Synthesis, and my degree is in analytical science, and I have a postgrad diploma in quality management, certified in GDP, pharmaceutical auditing, and I'm a qualified QMS lead auditor also. And Daniel is Daniel Kirschner. He's based in Cambrex in Durham. He's the Executive Director, Analytical and Biopharmaceutical Services at Cambrex. He has 15 years plus in pharma with 8 years in the CDMO sector, focused mainly on analytical R&D, method validation, and regulatory support. His expertise lies in analytical chemistry, method development, spectroscopy, chromatography, and mass spectrometry. His former roles at Avista Pharma, Gilead Sciences, and Nectar Therapeutics makes him an industry expert. He holds a PhD in bioanalytical chemistry from the University of Alaska Fairbanks, and I'm delighted to have him as my co-presenter today. So let's make a start. So the stability of pharmaceutical products really is a cornerstone of quality, and it ensures that all medicines remain safe and effective throughout their shelf life. Now, this has been governed by ICHQ1 since the early 1990s, and now we have a radical overhaul. So today we'll talk about the updates to the guideline, the key drivers of change, the implementation timeline. We look at the industry feedback from the EMA and the FDA, and we'll explore what companies need to do now to manage the updates and discuss the implications to the industry as a whole. So in this section, we look at the updates to the guideline itself. We look at the background, the key drivers of change. We'll explore what's new and look at the timeline for review and implementation. So the original ICHQ1 focused on the stability testing of new drug substances and products. And over the next decade, the supplementary guidelines were added to address more specific aspects, including the photostability testing, ICHQ1B, and new dosage forms, ICHQ1C, etc. So it has since been revised twice, with the most recent version being ICHQ1A revision 2, which was finalized in 2003, and that's the one that you're all familiar with at the moment. The 2025 overhaul is the most significant update to stability testing in over 20 years. It consolidates Q1A to F and Q5C into one single comprehensive guideline. So this new draft introduces a more modern and risk-based science approach to stability testing. And this is largely welcomed and is rooted in other ICH guidelines. For example, Q9 risk assessment and Q10 pharmaceutical quality system. And it also covers, this new draft also covers advanced therapies and stability modeling, which has been lacking really up to now in the previous version. However, you have to note there will be some implementation challenges ahead. So why the update? Well, the original guideline reflected the scientific and regulatory standards of its time, but the advancements in technology and scientific understanding and tightened regulatory inspections have now demanded a comprehensive revision. So it is prompted by the need to streamline and modernize stability requirements, address ambiguities in the previous guidances, and incorporate scientific advances and new product types. So this 2025 update integrates principles from the other ICH quality guidelines to reinforce the science and risk-based approach to stability testing. The ongoing advances in pharmaceutical science and technology call for a more flexible and a more contemporary approach to stability programs and testing protocols. The consolidation of guidelines. So the ICH new draft has merged the existing multiple stability guidelines into a single unified document. And this involves 18 individual sections and 3 annexes, and it covers the entire stability study process from early development all the way up to post-approval. And these changes help to make the guideline more logical and more inclusive. But one of the main aims of the update as a whole was to improve the clarity, consistency, and harmonization across diverse product categories. The update also tackles new technical challenges and includes stability strategies for ATMPs, advanced therapy medicinal products. Now, this is a huge update, and it reflects the growing importance of complex biologics and cell and gene therapies within the regulatory landscape. So what's new in the draft? Well, let's start with the structure. So the revised guideline is much more comprehensive. It now contains 18 sections and 3 annexes, spanning a massive 108 pages in total, which together provide detailed guidance across a wide range of stability testing topics. The expansion reflects the complexity of today's pharmaceutical landscape, which has changed since the original version was released. One of the biggest shifts is in the methodology. So the draft promotes a science and risk-based approach, and this encourages innovation while ensuring consistency in how stability is assessed globally. It's a welcome move away from the rigid one-size-fits-all models. For example, a uniform approach to the preparation of a stability testing protocol is no longer required, but providing all the decisions can be justified through robust scientific rationale and risk assessment where appropriate. There's also more clarity in key areas, offering expanded guidance on stress testing, storage conditions, in-use stability, and statistical modelling. And these are all critical for designing robust stability programs. And importantly, it addresses advanced therapies. For the first time, there's specific guidance for the ATMPs, and this includes cell and gene therapies. and these in their very nature present unique stability challenges which are now being addressed. It's a major step forward in aligning regulatory expectations with emerging technologies. The update introduces several content areas not fully addressed in the previous Q1 series. For example, it discusses the stability of novel excipients, vaccine adjuvants, and provides new guidance on in-use stability studies. This is especially of interest to ensure product stability during the period of patient use after opening. Short-term stability studies are also a welcome addition to address the issue of excursions during transportation or temperature storage. So this slide is just an overview of the progression of the ICH-Q1 stability guidelines since its original implementation. Starting in 1993, it was first revised in the early 2000s with the development of the supplementary guidelines, and this overhaul has been in the pipeline since 2023, this new one. It's now at stage 3 of the update process with the final draft expected to be released the end of 2025 and implemented in 2026. So why is this revision generating such chatter and interest within the industry, you may ask? So the revision is being presented as a major step forward in just how we manage our stability across the product lifecycle. So the updated guideline supports quality assurance from the development stage all the way through to post-approval, reinforcing the importance of stability as a continuous process and not just a regulatory milestone or a box to be ticked. It presents a holistic approach to stability testing, replacing the patchwork of documents, if you like, with a unified set of principles for all product types, small molecule, new chemical entities, biologics, etc. It provides a one-stop shop, nearly, for all stability requirements across the board. So the update to the Q1 guideline will help align stability practices with global regulatory expectations. And this is especially important for companies who operate across multiple markets. So here in Q1 Scientific, we offer support and assistance to customers to help them navigate the new guidance. So we interpret expectations and we advise them how to apply them effectively to their storage strategies. So compliance and consistency is key in successful stability planning and testing. And when implemented, this update should make the life cycle less complicated and the requirements clearer and easier to comply with, thus maintaining confidence in long-term storage and life cycle planning.
Nadine, a question that's come in earlier was about after finalization of the guidance in the scheduled period for review and revision. Do you know of any additional topic areas that are waiting in the wings that would be eventually added on to this, since it's been so progressive in some of the more recent areas where stability is being developed?
Well, there's a lot of discussion at the moment where the industry has called for specific examples of case studies on how to implement the leaner stability protocols that are allowed through this guideline. So that's going to be discussed later on through the presentation.
Okay, yes, quite impressive, the lineup of things that have been included in this one. It makes one wonder if there's going to be a continual waiting period to do that. Well, thank you for that.
All right, talking a little bit about modeling and analytical perspective, from Cambrix's analytical testing perspective, this revision really opens up a lot of exciting opportunities for innovation, particularly with how we approach identifying and controlling critical quality attributes and making science-focused decisions for supporting our short-term and our long-term shelf life studies. This guidance, it enables science and risk-based strategies, as was mentioned, and that really means we can design smarter and more efficient studies, and those studies can be backed by data. And that's a common thread in recent revisions of ICH guidelines, which are really focusing on harmonizing science-based quality. Bracketing and matrix studies, which reduce testing burden for a lot of drug products that have different strengths or dosages, or fill contents, those are discussed in detail in the new draft. There's also an entirely new annex dedicated to modeling stability data for predictive shelf life, including some advanced modeling. And what that means is for example, if we have a pharmaceutical with well-defined critical quality attributes and an established degradation profile, then we can really capitalize on the enhanced stability modeling approaches within the framework of this guidance that is now allowed to back us up. And we can verify and validate our models. And that's really going to change the way our studies for pharmaceuticals are designed. And this is going to be a big win for the pharmaceutical industry in general. And it's going to lead to leaner protocols and reduced testing, particularly in the commercial small molecule settings. There's also a lot of clear guidance that's been added in stress testing and use stability and reference standards. And that allows us to better predict our shelf life and to optimize our testing. And each of these sections on these topics provides framework that spans small molecules and biologics. The new guideline also has a framework for the pretty complex world of advanced therapeutic medicinal products, the ATMPs. These are really a diverse category of biologically derived treatments. They include tissue engineered products, cell and gene therapies, and many viral and nucleic acid based treatments. And these products, they really span some of the latest cutting edges in advanced treatment options. But they're a lot more complex than just about all pharmaceutical types from past guidances. Some of these products even have multiple biological mechanisms of action. And so as a result of that, that makes defining critical quality attributes for these ATMPs even more difficult. So a lot of ATMPs don't follow traditional approaches for ICH stability studies. So performing a traditional accelerated study, it can be of less value and less predictive than a more tailored study focused on real-time shelf life under intended storage conditions because of how some of these ATMPs are very fragile. Additionally, this new annex on ATMPs, it focuses on how to approach them for designing your studies, which may require quite a bit of customization and risk assessment. Some may be very small custom batches or may require advanced analytical techniques to assess cell viability or potency. So we've, for example, in our lab, seen live cell impedance assays for viral-based products that will test a specific active or aspect of the drug's activity, essentially an in vitro test, and that's used as a measure of the potency of the drug. And that's often just one of multiple techniques that can be used to assess the potency or viability of an ATMP. Additionally, this guidance discusses some of the relationships between some classes of ATMPs and their starting materials or intermediates. And that can require some additional consideration as well, because a lot of starting materials may be a cell bank or seed banks, and often the stability of those originating banks can be linked directly to critical quality attributes for the final product.
Daniel, we have a question.
Yeah.
How AI/ML will work on stability modeling? How
AI/ML?
Yes, is that typed correctly? That's from Suresh Patella. Suresh, if you want to elaborate a little bit more about AI/ML in the chat.
Yeah, this is Suresh. The recent guidance, including that AIML will be integrated to design the stability modeling. That's what we are wondering, like how we can do that. Even I was gone through some kind of literature, and some kind of algorithms are used to do that. But we're very new to that because this is a tech. We are not technical background from the software kind of algorithms. We are not aware. So how we can integrate that to design the stability modeling?
When you're referring to AI/ML, are you talking about like artificial intelligence modeling approaches?
Yeah, you're right.
Okay, got it. So I could speak to my knowledge on this. I mean, as more of an industry SME in analytical chemistry, I think artificial intelligence being used in modeling could be extremely powerful, and I think we'll probably see that over the next, 5 to 10 years, depending on the companies and their extent of validation of some of those processes. I think for me, what it really comes down to is being able to create a model and then be able to validate that model. And that's written into these guidelines. They really want to make sure that if you do come up with a model for for reduced stability testing, then you're going to validate that model. And so if you can do that with an AI-based platform, especially for some of the compounds that may be out on the market and simpler, I think, that would be great. And just making sure you can validate that model.
Yeah, just to jump in there, I'm seeing that we did an article on AI and what impact might be on stability. I'm seeing someplace in the future, because it's going to take a while for all the regulatory agencies to get comfortable with this, where we will have the reduced protocols and less testing based on advanced data and data analysis on previous batches. So I think it's going to work out, but I'm not hopeful that we'll be seeing it anytime in the next year or so in widespread use until we really get our hands around it and can prove what goes on with the regulatory folks.
Excellent. All right, so just wrapping this slide, I'd just say, it's ultimately really just about bridging the regulatory expectations with technical execution and ensuring that what we do in the lab is going to align with what the regulators are expecting at the submission.
Okay, so we move on to the next section where we look at industry reactions and EMA and FDA feedback and what they've said about the update so far. So the draft was originally published in April 25, and it was open for industry feedback until the end of July. So Q1 Scientific recently released an article reviewing the comments from EMA and FDA industry on the proposal. Now, this article was more like heading straight to the comment section of a trending story, if you like. It captured the real-time feedback from webinars and conferences, social media, and it formed a snapshot of what the people are really thinking. So let's review the positives first. So overall, the mood appears to be cautiously optimistic. To be fair now, it'd be difficult not to see the draft as a big step forward, especially in the consolidation of Q1A to F and Q5C into one unified document. It simplifies and harmonizes expectations across all product types. The modular structure is praised for being more usable and more comprehensive. And what's especially good is the incorporation of the quality by design QBD concept into welcome update. Now, in the context of the ICH revision, this QBD refers to a systematic science and risk-based approach to pharmaceutical development and lifecycle management. It enhances product understanding and encourages the tailoring of stability studies based on the nature of the drug substance or drug product, its formulation and packing, and its environmental sensitivities. So it's no longer a one-size-fits-all approach. Instead, there's some welcome flexibility, allowing the design of leaner stability protocols. But again, once the justification is scientifically sound and appropriate risk assessments have been carried out where required.
There's also some strong support for the inclusion of the ATMP specific guidance and predictive modeling. These updates reflect modern science, and they address a lot of real-world challenges. And the shift towards risk-based planning and flexible study design is seen as progressive, and it aligns with QBD principles. So these principles, they're the basis for a lot of these changes that have occurred in the recent ICH guidelines.
So let's look at the areas of concern then. So despite the optimism, there's still some concerns around clarity. Some sections still feel open to interpretation, which could lead to inconsistent application across various regions. And the documentation burden is a recurring theme in the feedback. Companies will need to justify their stability strategies much more thoroughly, and that could be a tough ask for smaller teams who have limited resources. There's also a need for training and change management. The new structure and the new methodology will require time and resources to implement effectively. And again, it could pose a significant challenge to companies.
And finally, some are still worried about regulatory conservatism and how that might limit the flexibility the draft is really aiming to promote, especially around leaner study designs. So while the guidelines provide this foundation, there's some authorities may have differing ideas on how much flexibility they think should be allowed. Also, there will continue to be some uncertainty on how this guidance, as it rolls out, will impact programs that are in flight, and that's going to take some time to sort out. So let's take a look at how regulators are responding to the draft ICH Q1 guidelines. So while Q1 Scientific captured an informal industry sentiment, kind of like scrolling through the comment section of a trending story, that EMA and FDA feedback, it gives us a more structured and strategic view. And there's some quite positive support. Both agencies strongly support the consolidation of legacy guidelines into a single and modern document. And it's seen as a major step towards global harmonization and regulatory clarity. There's also clearly enthusiasm for a shift to science and risk-based approaches, especially as it pertains to life cycle stability management and flexible study designs. And the EMA specifically endorsed Annex 3, which addresses the ATMPs, but they're also requesting more detailed guidance to support areas like decentralized manufacturing and platform technologies. The FDA, their feedback has added some depth in areas like statistical modeling, in drug device combinations and vaccine stability, showing a strong interest in practical implementation.
Well, let's look at the key recommendations and concerns. So regulators are asking for clearer criteria for the reduced stability protocols that will be allowed. Say, for example, what's the minimum data set needed before companies can apply them? There's a call for harmonized standards around predictive modeling, including validation requirements, confidence intervals, and use of historical data. Now, both EMA and FDA agencies call for more practical examples and case studies to help companies interpret and apply the guidance consistently. The FDA feedback, it also highlights gaps in the guidance for drug device combinations and also for temperature sensitive vaccines, which are now becoming increasingly common. So the key friction points are clarity in terms of definitions, what constitutes sufficient justification, feasibility, how easy is it going to be to implement, including data, infrastructure, cost, etc., and transitional arrangements for legacy products and existing study data. So in summary, the feedback shows strong support for modernization, for sure, but also a clear need for clarity, consistency, and practical tools to help the industry implement these changes effectively.
Right, so let's talk a little bit about some of the anticipated changes. This slide outlines what we might expect in the final version of the ICH Q1 guideline, but it's important to note that this is an estimate based on feedback from industry stakeholders, from regulators, and from expert groups. The feedback is gathered during public consultation period, including formal input from the EMA and the FDA. It gives us strong indication of where the guidelines likely to evolve. We're seeing calls for more detailed guide areas like ATMPs, predictive modeling, reduced stability protocols, and that really reflects the industry's need for clarity and flexibility in applying some modern stability strategies.
There's also a push for practical examples, harmonized terminology, and alignment with other ICH guidelines to support consistent global implementation. There does appear to be some concern over the consistency of definitions, for example. So in the final draft, we would expect to see extended guidance for ATMPs, including decentralized manufacturing and platform technologies, clearer criteria for reduced stability protocols, including the minimum data set, for example, greater regulatory flexibility for changes within the pharmaceutical quality system, enhanced statistical modeling guidance, including validation standards and acceptance criteria, improved clarity on container closure systems with bracketing and material-specific examples. More practical examples and case studies are called for to support implementation and harmonized interpretation. Additional support for combination products and vaccines, especially drug-device interactions. Alignment with other ICH guidelines, for example, Q5C, Q12, to promote global harmonization. And recognition of digital tools and predictive modeling with clearer expectations for validation and use. So while the final guideline is still under development, these themes suggest a strong move towards science-based, life cycle-driven stability planning with better support for emerging therapies and technologies. So while we wait for step 4, it's a great time to start preparing, reviewing protocols, training teams, and identifying your gaps in the current practices.
Thanks, Aideen. Let's hold there for just a moment. We have some more questions coming in. And just before we get to those, I'd like to do the ubiquitous infomercial. Halfway through our time today, we'd like to take this opportunity to encourage those of you not yet familiar with StabilityHub to visit us at stabilityhub.com and explore the site for features that are most useful for you and to your team. And sign up there, if you would, for the monthly stability newsletter, which included the article that we're doing a webinar on today. Please encourage your colleagues to do so as well. You could do us a great administrative favor right now if we take a bit of a poll. If you're attending today because you saw our notice through LinkedIn, please raise your virtual Zoom hand so we can get an idea of how effective that route is. And just leave it up there for a while. We'll take the count while the presentation continues. If you enjoyed the Stability Hub webinars and the give and take style of discussion and sharing of stability information, you might want to put May 17 through 19, 2026 on your calendar for an extended opportunity to network and benchmark with your stability colleagues in person. Our pharmaceutical stability discussion group will be meeting in North Carolina. Details will appear in the events section of the Stability Hub site. We're seeking companies that might also be interested in hosting such meetings in the future, bringing the best of the stability world to their own campuses. Contact us through stabilityhub.com/contact for more information. And now back to our program with the question submitted about protocols. Let's dial this in here. For primary stability studies below 12 months shelf life drug products, testing to be done every month up to 3 months and every 3 months after 3 months up to 12. How can we deal with it? So let me do a little interpretation. I know you guys are thinking about that. You can pop in. We also now have Sherilyn Campy with us. And if Sherilyn, if this pops into your realm, you're welcome to comment as well. Primary studies below 12 months. So we normally do every 3 months. And we're thinking the new guidance will eventually make it so that we don't have quite so many points.
Yeah, I think what I can say about this is the time points for a protocol should be based on the stability profile, the stability of the molecule itself. And I believe that the ICH, the draft of the ICH actually speaks to that. I think I remember the section that you're referring to about the frequency of time points based on the shelf life. And there is some verbiage in there that attention should be paid to, or you should consider the stability of the molecule when you design the protocol and the frequency of the time point. So I would just guess that if the shelf life is 12 months, it's likely that, that the product may not be stable beyond that, right? Or that it'd be less stable. And so make more sense to collect data more frequently. However, if the product is stable and it's because, that is all the data that we have at this point prior to submission, again, I think that you could use supporting data to get around that. But again, I think that the idea here with the ICH draft is to use science and risk-based approaches and use the data on hand, use your development data to be able to support, the design of your stability protocol. So these are all recommendations that are provided in the draft of the ICH. And again, if you have data to support or get around it, I think you could probably present that as well in the filing.
Thank you, Sherilyn. I should mention Sherilyn served on some of the committees that fed information and feedback back into this particular guidance, and that makes her a very valuable asset for us today. Any other comments on the shortened protocols less than 12 months? If not, we can continue. Looks like we can go ahead.
All right, move to the next slide.
Okay, I'll drive on. Okay, so we just look at this section on strategic implications. What might this new guideline mean for your stability program, and how might regulators interpret these leaner protocols that have been suggested?
So this section, it's all about what the draft ICH guideline could mean for your current stability programs and how to start thinking strategically. First, companies are going to need to review their protocols. The draft really encourages a shift towards risk-based and lifecycle focused approaches, and that's going to mean stability planning should be proactive and continuous and not just a regulatory checkbox. There's also here a real opportunity to streamline your study designs using tools like bracketing, matrixing, and modeling where it's appropriate. And those approaches are now formally supported in this draft, but they need to be scientifically justified to gain acceptance. And with that flexibility in design, it comes some responsibility. This draft, it places greater emphasis on documentation and justification, especially for leaner protocols. So regulators are going to expect clear rationales and supporting data. And that's why doing a gap analysis is so important. Companies should start assessing where their current practices diverge from the draft guideline and identify areas that may need to be adjusted even before the final version gets released. If it's implemented well, the changes, they should reduce testing burden. They should also improve efficiency, but really that's going to depend on regulatory confidence in the science behind your approach and how consistently it's interpreted across the different regions. So the key takeaway here is, start preparing now. The final guideline, it may still be evolving, but the strategic direction of it is very clear, and the companies that act early, they're going to be better in a position to adapt.
We have a question that's come in as well. It pertains to, I think, to a little bit of earlier slide content. Can the presenter summarize any feedback on the revised guidance from other regulatory agencies, in particular ANVISA and TGA, as they have historically presented greater challenges in the registration of biologics? If you need me to reread, I can.
Yeah, reread. I'm also trying to think if I can answer that question. So,
did you have any comments or ideas about feedbacks that come in from the other regulatory agencies on this revised guidance, in particular ANVISA and TGA? So the Australian folks and Brazilian. And you're right there, as the comment goes here, it does represent greater challenges. They tend to come up with some pretty stiff barriers here and there for us to overcome.
Maybe one thing I could probably say, I don't have an answer per se, but I'm just kind of speculating. So the expert working group, that worked on this draft had representatives from various regulatory agencies. I have to go back and check if they also included representatives from ANVISA and, so I'm not really sure, but I do know that it spanned, regulatory agencies from globally. So, and then of course included industry reps as well. So, and this draft was worked on for a number of years. I mean, as we saw in an earlier slide, the concept paper was approved in November 2022. So over this whole period of time, I think it's been, all of the recommendations and the design and all of the information that went into this, included all of the feedback from various agencies as well as companies globally. So just want to make that statement.
Okay, and I'll point out that our conversations through Stability Hub and the Pharmaceutical Stability Discussion Group never really end. There's always new information coming out, more people asking follow-up questions. So I've taken down some notes here, and we can pursue this online with our website. And as soon as we get further information, we'll make that known to everybody. The Stability Hub discussion forum is always available, and you can ask questions there for people to answer and try to get some information to you. So it looks like we're ready to move further.
All right, next slide. All right, so we're going to talk a little bit about impact assessment. What we have is a quadrant table here to kind of look for areas where you can have a really strong impact or low impact in how you respond. So we'll take a look first at the top right quadrant. This is your high impact, high complexity. These are the must-win battles. They're going to require the largest investment in your time, in training, and in generating the data. But they're also going to be the highest regulatory risk if they're mishandled. And so an example of that would be if modeling is not justified properly, your regulators may reject the proposed shelf life, and that could cause a delay in your launch. In the top left quadrant, we have some of these high impact, low complexity tasks, and these are going to be your quick wins. You can get them done early to de-risk, and an example of that would be like updating stability protocols for Zone 4B, storage conditions, and that's mostly a procedural exercise. Recommendation would be start those immediately, as these changes are relatively inexpensive, but they'll allow you to be compliant. The bottom right quadrant, that's your low impact, high complexity. You should approach that selectively. Just invest in this when it makes sense for your company. So an example of this would be the annex for ATMPs. It's going to be irrelevant to some companies without cell and gene therapies. So monitor this, but don't overinvest unless it's really applicable to you. And then the bottom right quadrant, that's going to be your low impact, low complexity, your housekeeping tasks. And you're going to do those in parallel as part of your routine quality management system updates. So this could be like aligning some of the new terminology that's being used, updating your templates. So you could bundle that up with your normal SOP review cycles and minimize the overhead that it's going to take to implement those changes.
Okay, so let's shift focus to how regulators might respond to leaner risk-based stability protocols. So the draft ICHQ1 guideline, it opens the door to streamlined study designs, things like bracketing, matrixing, predictive modeling. But the big question is, how will regulators interpret these new approaches? So feedback from the EMA and the FDA shows support for flexibility, especially when the changes are managed through the pharmaceutical quality system, and that's encouraging. But there is a note of caution, because some jurisdictions may be more conservative, especially when it comes to reduced protocols. Without clear justification and robust data, leaner designs could face pushback. So that's why scientific justification is key. So regulators will expect companies to explain the rationale clearly and not just in theory, but with supporting evidence. The final guideline may include case studies or examples to help standardize interpretation. But until then, companies should be prepared for variability across regions. So the takeaway here is this: leaner protocols are possible, but they must be defensible. Engage with your regulators early, clarify expectations, and avoid compliance issues. Document everything thoroughly and be ready to adapt based on feedback received. Remember, this update is new for regulatory agencies too, and they might need time to accept the new approach also.
All right, we're going to talk a little bit about preparation, what you can do now, gap analysis, planning, and training. So what companies can do right now to prepare for ICHQ1 guideline? First and foremost, read the full draft. It's large. Again, I think it was mentioned about 108 pages. It's very comprehensive, 18 sections, 3 annexes, and you need to understand its structure and understand the intent. That's really key. Some of the content may be placed in different places than previous guidelines, so read it all the way through. And then conduct a gap analysis and to compare your current stability practices and SOPs with the draft requirements. And that's going to help you identify where some updates might be needed, whether it's in documentation or whether that's how you design your studies, and evaluate your data. And then, review your stability protocol and ask yourself a question. Do they align with the new risk-based lifecycle driven approach? Are you using tools like modeling or bracketing? Are you doing matrixing? And are they scientifically justified? It's also time to start planning your training sessions with your teams. And that could be from QA to regulatory affairs. They're going to need to understand what changes are occurring and how to implement them. Also, don't forget to engage with the regulators and get that clarification to help avoid compliance issues later. This final guidance, may still be in development, but there's opportunity to prepare, and that really starts now.
Just to summarize our presentation today, we'll have a look at resource sharing and just sum up our key points and look at some questions and answers. So when we wrap up, let's reflect on what this guideline revision really means. Development, the final one won't be in place until the end of 2025 at the earliest. But the direction it's going in is pretty clear, and the opportunity to shape your future compliance strategy starts now. It isn't just an update, it's really a strategic shift in stability testing. The revised guideline introduces a modern, risk-based and lifestyle-driven approach to stability testing. It's designed to support innovation while maintaining regulatory confidence. Now, industry feedback has been largely positive, especially around the consolidation, modelling and the ATMP guidance. But it also highlights the need for clarity. We need consistency and we need readiness, particularly when it comes to the interpretation of leaner protocols. Companies need to prepare for regulatory variability. Not all authorities may interpret the flexibility in the same way. So scientific justification and documentation will be key. Now for Q1 Scientific and for Cambrooks, this is our moment to lead by guiding customers, advancing analytical strategies and helping the industry navigate this new era of stability science. So I just want to highlight the key resources that helped us draft today's presentation. So firstly, we have the ICHQ1 Step 2 draft guideline and the official Step 2 presentation, both of which published by the ICH Expert Working Group. These documents outline the proposed changes and provide context for the revision process. Next, the EMA feedback summary, and this offers insight into how European regulators and stakeholders are responding to the current draft, including support for ATMP guidance and calls for more clarity in certain areas. The FDA public comments are also available online at that website. And these reflect a broad range of industry perspectives, including detailed recommendations on modelling, combination products, and life cycle management. And then finally, our own Q1 Scientific article published recently. So it provides a snapshot of more informal industry sentiment, capturing real-world reactions from all the webinars, conferences, and client interaction that we have had. And it has helped us to understand how the draft is being received by the people on the ground. So together, these resources offer a well-rounded view of where the guideline is heading and how companies can currently prepare. So how can Q1 Scientific and Cambric support you through this? So for Q1 Scientific, our aim is to guide our customers through evolving expectations for storage conditions and life cycle planning. We share our expertise on real-world implementation of the final guideline, and we continue to be a trusted partner in GMP and GDP compliant storage across global markets.
And from Cambrix's perspective, on the analytical testing side, our focus is identifying your critical quality attributes for your drug substances and your drug products and helping develop those control strategies. So we really lead the way in method development, validation of modern stability programs, and that can apply to any stage of clinical development. We also provide some key technical support for designing your studies, and we collaborate with regulators and with the industry to really shape science-based compliance solutions.
Okay, so before we close, I just want to share an important event for anyone involved in stability science. So Q1 Scientific hosts its 4th annual Stability Studies Conference on October the 22nd to the 23rd, 2025 at the Killashee Hotel in Naas, which is in County Kildare in Ireland, beautiful part of the world. And this conference will bring together industry experts, regulators, professionals from across pharma and medical devices, and it will explore challenges and innovations in stability studies. So in this conference, you can expect a mix of presentations, panel discussions, networking opportunities, and these are all focused on managing and evaluating stability studies across the product lifecycle. So whether you're in quality, reg affairs, analytical testing, or stability storage, it's a good chance to gain insights, share experiences, and prepare for the future of stability science as it continues to evolve. If you need any further details, visit our dedicated conference website. Well, thank you so much for joining us today, and thank you to StabilityHub for hosting this webinar. Yeah. We really appreciate your time and engagement throughout the session. So if you have any questions, now's the perfect time to ask. Feel free to drop them into the chat. And if you don't get to your question today, or you think of something later, that's no problem. Feel free to follow up with us afterwards.
Thank you, Q1 Scientific folks. So we are open for questions for several minutes, and a few that came in advance, and this might be up Sherilyn's alley. Now that the review period has closed, are there ways to amend or improve the guidance as it moves through further stages, or is it totally locked?
So you're right that the review period has closed, but the guidance is not final yet, right? So there are still a lot of comments that, industry has provided for one, and then I know I sit in I'm a member of the stability community for AAPS. I know through the regulatory response team at AAPS, we had like a number of comments that we sent out. So there's various groups that are sending comments in, and the EWG will take all these comments under consideration, and so it's not final yet. And we'll see. Hopefully, it will be more refined, more clear on some of those questions that we have, and yeah, we'll find out in a number of months how that works out. But it is not final, and there isn't sort of like a periodic review type of period like we would do for an SOP. That is not the process, but there are ways of providing feedback on the ICH once it does become final. I think if you go into ich.org, there are various mechanisms of doing that through reflection papers and so forth, providing that additional information that might eventually turn into like a future update of the ICH.
Thank you. Follow up to that, are any similar plans to consolidate guidances dealing with topics such as SUPAC and some of the other ones about post-approval? Anything along those lines?
So this is to consolidate the SUPAC guidelines. Is that the?
I'm just asking if does it covers things along the lines of SUPAC. I didn't notice it on my passage through there.
I don't know if there is a plan to consolidate guidances on the SUPAC, but keep in mind Q1 has a whole section on SUPAC. There's a lot of references to this is scale up post-approval, right, commitments. So there are a number of references to that, a lot of guidance in ICH Q1 on that overall approach. So that's what I can say.
Okay. Thank you. I'm looking to see if we have other questions popping up in chat. Other than that, we have some pre-submitted ones. This one might be of interest to our Q1S folks. What should we do if we're mismatched with our CDMO, if they are way ahead of us or way behind us, if implementing provisions of the guidance? So if your partner going one direction or another is lacking in getting the guidance implemented or starting to follow it in advance, what's the best route to do? Do they need to drop back and do more planning, or do you need to kind of dumb down your program to stay back with them?
So maybe I'll start this one off or take this one, but I would say that every CDMO is different, and I've worked with a lot of different CDMOs. We focus a lot on really trying to build a partnership, a regulatory partnership with our clients, and so we're going to help guide them through any guidance where they need to be compliant. So if we see something that they're doing that doesn't look like it's going to be compliant, obviously our SOPs are going to be updated appropriately. So if we're at the advanced state and we're not where and they're not where we're at, we're going to obviously lead them there because all of our procedures are going to be built in that advanced state. Now, if it's the other way around and we have clients that come in that are further along than we are, we really have to take an adaptive strategy to figure out how we're going to bridge with them. And so I think it just depends on the CDMO and their focus areas. But I know companies like Cambrix, like Q1, stay pretty adaptive and can flex in either direction.
Okay, thank you. Has anyone identified any impossible aspects within the guidance? In the comments, just saying this is absolutely not doable. I didn't see any of that in any of the commentary that I've read online or in this presentation. I didn't know if anybody had a stop the train, I want to get off kind of objection.
John, I'll just take that one. I don't think any aspect is truly impossible per se, but there's no doubt that the guideline does introduce some significant hurdles for companies. The smaller organizations with smaller stability teams and limited resources, it's going to be a challenge to train and bring in the new protocols using change control, and maybe or maybe not they'll have the expertise on site to do that. Some of the companies will be navigating various different multi-jurisdictional issues. So will all regulatory bodies have the same approach? So that has to be yet determined.
Okay, thank you.
I will just add one more thing. I did not feel reading through the guidance that any of that information, the content is new, particularly new. I think they were either spread across various guidelines, regulations, or were more like expectations, right? And so a number of years, the expert working group was, again, I'll go back to the composition, included regulatory agencies from various countries, as well as industry representatives. And so the content really reflects the expectations and regulations that have been around for a number of years. So hopefully there is nothing new, and it's just now contained in one place, essentially.
Well, thank you. We have to bring our program in for a landing here because it's approaching 1 o'clock very quickly. And as we bring it to a conclusion today, we invite you all to join us on Thursday, October 23rd at 11:15 a.m. Eastern for the Pharmaceutical Stability Discussion Group office hours, which is a 90-minute opportunity for you to bring stability questions, challenges, and war stories before a widely experienced panel of stability subject matter experts. No presentation on that day, just answering the questions. Whether you have questions or just want to pick up some pointers, you'll definitely come away with some valuable information. Return on Thursday, November 20th, when parameter generation and control will bring us vital details for recovering from stability chamber excursions with respect to both equipment and product impact evaluations. And in December, Novatech will be back with their final installation of stability management digitalization. Watch for details in the monthly newsletter and in the events section at stabilityhub.com. I'd like to thank our presenters, Aideen Lin and Daniel Kirshner, panelists Sherilyn Campy and Walter Routh, and you, our great audience, for your attendance today. We hope to see you again soon. Thank you all.